In the picture of a post-meal situation where you have a spike in blood glucose levels, this spike is detected by pancreatic beta cells, which release insulin. So the insulin is going to be released into the bloodstream and detected by skeletal muscle cells and adipose tissue cells. So these guys are going to be in skeletal muscle and adipose cells. Now, this is a totally different class of receptor than we've talked about before. And what these guys are called receptor tyrosine kinases. So we can draw them kind of like these little bean-looking things in their extracellular domain, with a big transmembrane domain that extends into the intracellular side. So might not be the most elegantly drawn insulin receptor, but it's about what it's going to look like. So again, high glucose situation, insulin floats in and binds to the receptor. So interestingly, this receptor actually exists as two separate halves on the cell surface. But once insulin binds, it dimerizes the insulin receptor. The insulin receptor. So once this dimerizes, these receptors themselves are unique in that they actually have an enzymatic function, and in that they are kinases. So let's make a different color. Once these two come together, they will start to phosphorylate itself in what's called autophosphorylation. And being a receptor tyrosine kinase, these are going to phosphorylate specifically tyrosine residues. So if you might remember, your tyrosines are going to look something like this. Within a polypeptide chain, we have this phenyl ring hanging out on the side chain, and most importantly, we have an alcohol group, which can act as our nucleophile in this situation. So at the at this hydroxyl group, we can actually tack on a phosphate. So all of these guys on the receptor themselves are going to be phosphorylated at tyrosine residues. And what this allows is, now that we have phosphorylated residues, it allows the interaction of different associated substrates. So in the case of the insulin receptor, it allows for the binding of the ever creatively named insulin receptor related substrate 1, or IRS1. And this IRS1 is also phosphorylated. Phosphorylation seems to be the common language in cell signaling. And once IRS1 is phosphorylated, that again allows for the recruitment of another type of enzyme called phosphoinositol 3 kinase, or PI3K, which is often drawn like this. Phosphoinositol 3 kinase. So what this guy does is it'll take PIP2, which, if you might remember, is a phospholipid, and it's going to take this phospholipid and convert it, phosphorylate it to PIP3. So let's name this guy PIP2. And basically, we're going to stick another phosphate onto the sugary head of this phospholipid, and we're going to call this guy PIP3. PIP3. So, as you might remember, PIP2 is an important signaling molecule. PIP3 is also an important signaling molecule. And what it's going to activate, what it's going to signal, is an enzyme that kind of hangs out on the intracellular side, termed PDK1, or PIP3 dependent kinase 1. And what this guy's going to do, as most kinases do, is phosphorylate and activate another type of enzyme called AKT. And AKT is also called protein kinase B. So, by activation of AKT, our aptly named protein kinase B, as you might imagine, it's going to go on and phosphorylate a lot of different things. And the main outcome that we care about with the activation of the insulin receptor is through some long complex pathway downstream of AKT, we're going to upregulate GLUT4, the glucose transporter of high KM, high turnover rate. And again, I mentioned that these are in adipose cells and muscles, skeletal muscle cells. And by upregulating GLUT4, it serves as a way to take out all of this excess glucose from the blood and stick them either into glycogen stores or fatty acid stores or triacylglycerol stores elsewhere. So you might remember another type of GPCR, the GQ pathway, in which phospholipase C converts PIP2 into diacylglycerol and IP3, inositol triphosphate. So the reason that pathway, again, is important is because of a proposed mechanism of insulin resistance. So phospholipase C is a really broad... so downstream of phospholipase C, diacylglycerol activates protein kinase C, another very general kinase. So we often draw it kind of like this, protein kinase C. It's going to go phosphorylate a ton of different things. But if you get a buildup of diacylglycerol, again, just the fatty acid tail of PIP2 and PIP3, diacylglycerol can directly activate protein kinase C. So the way we get a buildup of diacylglycerol is through this high-fat, over-nutritious diet where we get an accumulation of triacylglycerols, which can easily be converted to diacylglycerol. We get the constitutive activation of protein kinase C. And what this is going to do is phosphorylate a whole lot of different things. But most importantly, we're going to phosphorylate the insulin receptor on say, serine or threonine residues, other amino acids with these reactive hydroxyl groups. So basically, we're going to get phosphorylation at places where we shouldn't be getting phosphorylation. And what that does is when insulin actually binds to this receptor, if we already have a huge amount of phosphorylation in various sites, this actually prevents this receptor tyrosine kinase from starting its autophosphorylation on the correct tyrosine residues. So if it can't phosphorylate itself, we don't get the recruitment of IRS1, we don't get any of this downstream pathway, and we don't get upregulation of GLUT4. So we maintain high blood glucose in this model of desensitization. I hope you guys found this useful. Good luck studying. I'll see you in the next one.